You're on. It started.
Okay, great. So welcome everyone to our second session for this undergraduate research symposium. My name is Cory Browning. I am assistant professor of French literature in the Romance Languages department. I'm happy to be to be the moderator of this panel. And one of the things I like about being a moderator at the undergraduate research symposium is it gives me a chance to really get out of my field. And so I'm happy to, in this case, be moderating a panel with largely in biology here entitled Inside Out. We have 5 presenters, and so I will introduce each of them and the title of their presentations as we go. And if you have questions, there is at the bottom of the screen a Q&A function. You are free to type in your questions during the presentations or to ask questions in audio format at the end of the presentations. Okay, so we'll go ahead and begin. Our first presentation, Carly Fear, will be presenting a presentation on computational modeling of BMP2 Affibodies. Carly?
Thank you. Let me go ahead and start. Oh, I don't think that works. Sorry. All right. Perfect. So today I'll be talking about modeling BMP2 binders. And this has been a project in the Hassanzadeh lab, and it's been a huge collaboration as well with the Hedy lab. So I actually started researching in the Ong lab, and I was working on bone regeneration and how it's affected by these mechanical signals. So what that means is when we walk or run, our bones are very slightly deformed. And this small physical signal is translated into a biological signal that promotes bone remodeling and healing, which is especially relevant when we're looking at fracture regeneration, and especially fractures that are unlikely to heal on their own because they're too big. So when I transitioned to start working in the Hassanzadeh lab, I was looking at bone regeneration, but instead of looking at mechanical signals, I was looking at biological signals. So the focus of my research is a particular signaling molecule called BMP2, which is bone morphogenic protein 2. And what it does is it docks to the outside of these cells in the region of the bones, and it causes a signal cascade that leads these cells to turn into bone-forming cells or osteogenic cells. And so BMP2 is a really important molecule to study. And what I've been looking at since my collaboration with the Hedy lab is how biomaterials can tailor the spatiotemporal delivery of BMP2 in these fractures that are unlikely to heal on their own. So we really like to look at these parameters of retention and release dynamics. So when we use a biomaterial like heparin microparticle or HMP, we see that there is both improved retention of the target molecule BMP2 in this region, which is shown by how there's sort of a yellow bright spot locally at that tissue, whereas in the absence of the biomaterial, this target molecule is allowed to diffuse into the surrounding tissues and is therefore not delivered as optimally. So again, retention and release are really important parameters of this system. And one instance where we see release that's non-optimal is this phenomenon called burst release. So down in this graph in the bottom right, there's a period of the burst release where soon after implementation, 6 hours after we add this material, we see that even when we're using biomaterial, there's a portion of BMP2 that's lost almost immediately in this system. And we really want to optimize this biomaterial usage to slow down that release and have better retention of BMP2. And this is important because when we get a burst release, this can cause a phenomenon called heterotopic ossification, which is the formation of abnormal bone or bone spurs during fracture regeneration. So when we use these biomaterials, the heparin microparticle, we see that we're able to inhibit the formation of these abnormal bone growths, but we still care a lot about this burst release. And so the project that I've been working with the Hediarachi lab on is their end of it is the directed evolution of Affibodies to bind our target molecule BMP2. So they have a large library of mutants, and they go through several rounds of selection to get Affibodies that will bind to BMP2 and make these interactions, which are shown in this figure on the right. And so we're able to detect experimentally that these Affibodies are binding to BMP2, but the problem is that we don't know how. And the mechanism of binding is really important for the functions of these Affibodies. So BMP2 is this dimeric protein in the middle, and it has two key regions where binding occurs. These are called the wrist and the knuckle. So the wrist is this pocket and the knuckle is this edge. And this is where its receptors bind. The goal with designing Affibodies is to have them interact with BMP2 specifically at these two regions in order to competitively inhibit the interaction of BMP2 to its receptors and slow release and signaling. So where I come in is the computational protein modeling and design. And so we use computer models to first model BMP2 binding of these Affibodies from directed evolution at the Hediarachi lab, and then use what we learned from modeling to re-engineer a set of Affibodies with a range of binding affinities at the wrist and knuckle binding regions in order to develop a tunable release strategy that leverages these binding affinities of the different Affibodies that we're using. So the basis of protein modeling is that it helps us approximate protein stability and therefore the occurrence of a particular conformation or interaction. The way that it does this is it quantifies the energy of a protein conformation or interaction in a term called score. And the lower the score, the better the energy. And so therefore, the more likely that a conformation is actually going to exist physiologically. And using these modeling platforms, we can go from predictions of structure to predictions of function. And this is really important work that I've been collaborating with the Rosetta Commons as well with, and their software has reached all kinds of different labs that are working on diverse problems to apply protein modeling and design. So that's been really exciting. Now getting into my results, I first modeled and predicted the structure of 4 Affibodies that the Hediarachi lab gave me and found that their structure is really regular. So they consistently fold up into these nice 3 helix structures, which is important to confirm that they're likely to have consistent functions as well. The next thing we wanted to do was approximate the binding site and orientation. So essentially how these Affibodies are binding to BMP2. And this confirms that these wrist and knuckle binding regions are really important, even when we're looking at rough guesses of docking. So these are 10 rough guesses that we achieved. And in order to further analyze the results from this docking, we wanted to look at the protein-protein interface and give it that score, that quantification of energy to see which interactions were favorable. So we got out 3 interactions that are favorable, 1, 4, and 8, which are shown here in this structure to bind at the wrist and at the knuckle. And so this is really exciting because it shows us that our predictions might actually develop into how we might form this tunable release strategy. So the next thing we wanted to consider is that our predicted structures, what I'm showing up here, these are snapshots, but really proteins are dynamic molecules. So they're constantly moving, and these movements affect their functions. So in order to study the movements of these proteins, we generated ensembles of the protein conformations, which are basically wiggled structures that look at how these things might be moving. And we found when we generated these ensembles and analyzed them that the wrist conformations differ from the knuckle conformations. And so since the scores are much lower, these wrist conformations are much more likely to occur in this particular Affibody interaction. The next thing we wanted to do was mutational analysis, which is looking at how substitutions of amino acids affect the Affibody BMP2 interaction. So this heat map is showing the effect on shape complementarity, the geometric fit of the two protein components to one another. When we substitute every amino acid in the Affibody for every other amino acid. So we got a couple patterns from this heat map that showed us that some specific positions or residues of the Affibody contribute to binding more than others. And we call these hotspots because they reveal important interactions between the Affibody and BMP2 at that interface. And confirming with the structure, we saw that these three locations or positions on the Affibody are clearly making direct contact with BMP2. So the future directions of after we've done this modeling work is to look at how these Affibodies are structured and to try to look at de novo protein design and making something from scratch. So what I'm showing here is two scaffolds that we've used that are completely not seen in nature. And we found that we can design these to bind specifically at the wrist or the knuckle. And during this summer, I'm really excited to start expressing and characterizing my designs and then seeing how they bind to BMP2 and what their affinities are in particular. I want to really acknowledge my lab. This is my lab group, the Hassanzadeh lab, and our collaborator, Marian Hediarachi, and her grad student, Jonathan, who's also been a huge part of this project. This has been such an exciting and interesting project. And so I really want to thank you all for coming to my presentation today and take any questions.
I had one question. You showed a graph earlier that was kind of a bar plot of the different scores for your Affibodies, and you mentioned that you designed 10 variants, but only showed 8 on the plot. So I was wondering if there were like potentially 2 that had like such high scores that they weren't interesting, or like what the disparity was there, if maybe I misinterpreted.
This one?
Yeah.
Oh, right. Okay, so these 2 in the middle, you can't actually see it because their scores are essentially 0.
Oh, okay. That makes sense.
Good question, though.
Other questions? As I said at the beginning, you are also free to type your questions if you prefer that method in the Q&A box, which is located at the very bottom of the screen.
I actually had a second question, if no one else does. One thing that I was wondering is whether Affibodies, because of their size, if that makes them kind of easier to implement rational design methods on and more like amenable to that process versus like directed evolution methods, or if their size makes them more difficult.
Right. That's a really good question. So they are relatively small. Affibodies are 58 amino acids long, and they contain these 3 helices. So their regularity makes them actually quite easy to look at, because we already know that there's a subset of the amino acids that are pretty much just contributing to that core structure, holding the 3 helices together. And there are only 13 amino acids that usually contribute to the variability of these structures and their interactions. So I would say their size definitely helps contribute to the ability to do rational design. Mm-hmm.
Okay, thank you for the questions. For the sake of time, for the further presentations, I'll ask us to hold the questions until the very end after everyone has had a chance to present. We do have a full schedule here, and we'll open it up to questions to all of the panel participants. Okay, so our next participant is Nora Kearns. She will be presenting her project on orthogonal system for the continuous directed evolution of genes in vivo. Nora?
Thank you. Okay, so my project is an orthogonal platform for the continuous directed evolution of genes in vivo, and I'm an undergraduate in the Kaelin-Pleasure lab. So why do we evolve genes? Well, genes are the genetic material which encodes proteins, and proteins are molecular machines which perform a lot of useful functions in our bodies and in nature. As an example of this, this protein here is globin. In our bodies, globin arranges into a larger structure that transports oxygen from our lungs to our tissues. Protein engineers recognize that these natural machines are super useful and we can modify them to perform novel functions in medicine and in industry. So the protein that I just showed you, globin, can be edited with 3 amino acid substitutions such that it can synthesize a precursor to the pharmaceutical ticagrelor, which is a medication for patients with heart disease. And this is really useful because normally the chemical synthetic pathway to arrive at this compound generates a lot of toxic waste. So if we can design biocatalysts that perform the same reaction, that helps us develop methods for greener pharmaceutical synthesis. It's impossible to talk about protein engineering and directed evolution without talking about Frances Arnold, who pioneered this method. As an example of her early work, she took an enzyme called cetylicin, which normally loses its catalytic activity in the solvent dimethylformamide. She took the backbone wild type gene for cetylicin and then performed sequential rounds of random mutagenesis on that gene. And after 6 generations of mutagenesis, she arrived at a variant which was as active in DMF as it was in water. And this kind of brings me to the reason we need to edit proteins in the first place is that proteins have evolved to function optimally in a host cell from their native organism, and they frequently lose function in a new context, whether that's a bioreactor or a foreign host or a novel solvent. Traditional approaches to directed evolution consist of rounds of in vitro diversification followed by in vivo screening and selection. So the gene encoding a protein of interest is mutated in a variety of different ways using any number of in vitro techniques to develop a library of differentially mutated genes. Those genes are then expressed in host cells, and the resulting proteins are screened for the select function. Typically, the best variant or the best few variants are selected, and their genes serve as the backbone for the subsequent rounds of diversification, translation, screening, and selection until an optimal phenotype is achieved. However, this process is really labor-intensive and time-consuming, and it necessitates a lot of work at the bench in developing these libraries and screening the variants. Continuous approaches to directed evolution allow mutation to take place entirely in vivo, so they circumvent this time-consuming in vitro mutagenesis. And they more closely mimic the evolution which takes place naturally within cells when those cells are exposed to selective pressures. So they rely on in vivo natural mutagenesis. However, this process takes place very slowly because host replication machinery, DNA polymerases, have very high fidelity and they mutate at a rate of about 10 to the negative 10th substitutions per base. But there are a couple things we can do to speed this process up, one of which involves using an error-prone host DNA polymerase. This error-prone DNA polymerase will introduce mutations to a gene of interest inserted in that cell, And over several generations, the gene will acquire enough mutations to let the protein, the encoded protein, evolve. But there's a problem here. This error-prone DNA polymerase interacts with the gene of interest, but it also interacts with the host cell genome. And due to the relatively high frequency of essential genes, these mutations will accumulate in the host genome and eventually kill the host. And this host is essential for replicating our gene and expressing protein. So if we don't have the infrastructure we need to do that, that limits the number of evolutionary generations possible. Directed evolution strategies aim to traverse sequence space at scale and depth. So sequence space can be visualized as this three-dimensional fitness landscape where genotype is represented by the X and Y axes and phenotype is represented by the Z axis. Scale, what I mean by scale is the ability to explore distinct areas of sequence space or even distinct proteins simultaneously, so engineering proteins at high throughputs. By depth, I mean the ability to access long mutational trajectories or complex mutations requiring potentially 20 amino acids to access. So these traditional strategies, which either require laborious in vitro mutagenesis or short experimental durations, limit access to both. So this brings me to my project, Diversify 29, which is an orthogonal platform for the continuous directed evolution of genes in vivo. By orthogonal, I mean two systems which do not interact. In my system, a gene of interest is replicated by a set of orthogonal replication proteins, which only interact and replicate the gene of interest without interacting with the host genome. This means that these orthogonal proteins, and primarily the Phi29 DNA polymerase, will only mutate a gene of interest without mutating the host genome. So this will allow us to evolve proteins over many generations without the host dying entirely within the cell. Phi29 comes from bacteriophage Phi29, which is a virus that naturally infects the host Bacillus subtilis. And what's unique about Phi29 is that it replicates by a protein-primed mechanism. E. coli replicates its genome using RNA primers, but Phi29 replicates its genome using terminal proteins, which are bound at the 5' ends of the DNA. So the Phi29 DNA polymerase recognizes this terminal protein to start replication. So we can recruit this system to establish orthogonal replication that doesn't interact with the genome. In our system, we have a linear plasmid encoding a gene of interest that's flanked by these sequences called ORIs, or origins of replication, that are necessary for replication by the DNA polymerase. This linear plasmid is bound by double-stranded binding proteins that melt open the origins of replication at a sequence that's recognized by the DNA polymerase, which then starts replication in a process primed by the terminal proteins, which then stay bound to the ends of the DNA. So again, this enables high mutagenesis on this linear plasmid encoding a gene of interest without increasing the mutation rate of the host genome. I now want to talk about some of the methods that I use to implement this system. I first had to construct a plasmid which carries the sequences encoding the Phi29 polymerase, and this plasmid also carries a kanamycin resistance marker. So I inserted this plasmid into cells, and then I screened for cells which successfully took up the plasmid by plating on kanamycin, which would normally kill the cells unless this plasmid is present. I then used a technique called polymerase chain reaction to identify cells which not only had the plasmid, but they had a plasmid which carried this gene of interest. PCR is a technique that we use to amplify just one specific gene of interest, and then we can validate length of that gene on a gel, which is a technique we use to check the length of DNA products. So I looked at colonies which had this length, indicating that the cell carried a plasmid with this insert in it, and then I sequenced that DNA to verify that it was the correct sequence. After inserting this plasmid, which I call P529, into my cells, I had to induce them with a compound called IPTG. IPTG activates a promoter which allows transcription of the downstream genes, the Phi29 proteins. So when IPTG is present, the Phi29 proteins are expressed, and we need these proteins to be around such that when PL is introduced to the cells, the orthogonal replication proteins can replicate the linear plasmid PL such that PL can then be transcribed and translated to the protein encoded within it. So I now want to share some of the results from my experiments. Induction of the Phi29 cells revealed that the Phi29 proteins are posing a metabolic burden to the host. We see this because the doubling time of cells carrying the Phi29 proteins is significantly longer than the doubling time of regular cells. So this suggests that cells are diverting metabolic energy away from proliferating towards creating the Phi29 proteins. However, these cells still demonstrated typical logarithmic growth. So even though they have slower replication times, they're not completely dying off, which means that the Phi29 proteins pose a metabolic burden, but they're not completely toxic, which is good. I then transformed my cells with a variation of this linear plasmid that carries the gene for dihydrofolate reductase R67, which is highly resistant to the antibiotic trimethoprim. So I could screen for successful replication of this linear plasmid by the Phi29 orthogonal proteins if my cells survived on trimethoprim. What I observed from my screen was that the cells are acquiring resistance to low concentrations of trimethoprim through some alternative mechanism that doesn't involve PL. So I screened on 2 different concentrations of trimethoprim, a low concentration, which is seen in the middle, and the high concentration, which is seen on the ends. And then my cells either did carry PL, which encoded trimethoprim resistance, or did not carry PL. So without PL is on the top row and with PL is on the bottom row. Ideally, what I would have seen is that when PL is present and IPTG is present, that means that the Phi29 proteins are present and PL is replicating. And those cells should have survived, these bottom 2 right here. But what I actually observed was that regardless of whether or not PL was present, at the lowest trimethoprim concentration, the cells survived. So this means that, because there's growth when PL is absent, the cells are acquiring resistance to trimethoprim through some alternative mechanism, likely by genomic mutations, which we refer to as escape mutations. So what could be going on here? Well, we know that this system actually works in vitro. If you just put the Phi29 proteins together in a test tube, they will replicate a linear piece of DNA. So this means that something is probably going wrong within our host. As I mentioned earlier, Phi29 is a virus which naturally infects Bacillus subtilis, and we're trying to force it to work in E. coli. Over evolutionary time, Phi29 has evolved its proteins to minimize deleterious interactions with Bacillus subtilis proteins, but it hasn't encountered E. coli in the wild, which means that these interactions haven't been mitigated or minimized. So there are likely some detrimental interactions going on within E. coli. However, we can leverage directed evolution to edit the Phi29 proteins to minimize these potentially deleterious interactions within our host, such that we can tune them to divert towards the linear plasmid and away from the E. coli proteins. Pending success of this project, we hope that it will allow us to explore sequence space at scale and depth. Because this is an orthogonal continuous directed evolution system, it will allow us to access long mutational pathways without resulting in host cell death. And because it doesn't require in vitro mutagenesis, mutagenesis takes place entirely within the cell, this will allow us to access scale. We don't need to develop in vitro libraries, so we can explore multiple proteins at once without this painstaking labor-intensive process of library development. I want to end by acknowledging some important contributors to my project, all of the members of the Pleasure Lab who have mentored me, particularly my advisor, Kaylin Pleasure. I also want to thank the Knight Campus Undergraduate Scholars Program for sponsoring my work, and the Undergraduate Research Opportunity Program for providing me opportunities like this to grow as a researcher. And now I'll take any questions, or I guess we'll actually save questions for the end of the panel. Thank you so much for listening.
Thank you, Nora. And yes, we will save the questions so we make sure that everyone has ample time to do their presentations, and then we'll take all the questions at the end. And so our next presenter is Phil Nosler, and Phil will be presenting his work on investigating a role for HSF1 in the formation of heat-induced DNA damage in developing sperm. Phil, it's all yours.
Thanks, Cory. Hi, everyone. I'm Phil. I'm a member of the Diana Labuda Lab in the Institute of Molecular Biology. And I've been working on a project to investigate sexual dimorphism in the HSF1-led heat shock response pathway. One of the interesting things about heat stress is that it's known to cause infertility, but specifically in males, not in females. This has been observed in humans and many other animals, and also even plants. And one of the significant things that happens following a heat stress is that DNA damage is induced in unusually high levels in developing sperm, but again, not in developing eggs. So a major question our lab is interested in pursuing is what's behind the sexual dimorphism of this phenomenon? Why are males and females responding differently to heat stress in regards to infertility and this DNA damage response? To address this problem, we utilize a model organism called Caenorhabditis elegans, which is a nearly microscopic transparent worm. It comes in male and hermaphrodite sexes, but the adult hermaphrodite only makes eggs, so for our purposes, it's a functional female. This worm has a few qualities that make it really great for genetic studies. It has a completely sequenced genome. It has a lot of available genetic methods for manipulation, and it also has lots of available antibodies and mutant strains. And also, as I mentioned earlier, it is transparent, so we can directly observe the development of sperm and egg cells without having to perform a dissection, which is an advantage. So we can use these qualities to directly observe what's going on in these worms following a heat stress. And what we observe is here we see developing sperm and egg cells with the DNA of each cell marked in blue, and all these little green spots are marking sites of DNA damage. And so what we see in the developing sperm cells is that there's a lot of DNA damage being induced following heat stress, but in the developing egg cells, we're seeing barely any. So not only do we have a really powerful model organism for studying, conducting genetic studies, but it's also portraying the same DNA damage phenotype and sexual dimorphism that's been observed in humans and other animals. So I'm particularly interested in investigating the molecular underpinnings of this phenomenon. And a great place to start is a particular pathway called the heat shock response pathway that is prominently activated following a heat stress in all eukaryotes. It's very widely conserved. And what this program does is it changes gene expression within the cell to promote stress tolerance and longevity as a response to the shock of heat stress. It's directed by a protein called HSF1, which is the master regulator of this pathway. And how this pathway works is HSF1 starts out in non-heat shock conditions as in an inactive state. And upon a heat stress, HSF1 becomes active and it enters the nucleus of the cell where it changes the expression of a wide variety of proteins of genes involved in a wide variety of different cellular functions. We're particularly interested in those genes that are involved in DNA repair and the maintenance of genome integrity, because these proteins could be playing, these genes could be playing direct roles in the DNA damage phenotype that we see in males after a heat shock response. So one of the first questions I was interested in asking regarding this pathway was simply whether HSF1 is involved in this DNA damage phenotype that we see in males. So over the summer, I produced some preliminary evidence as a part of the Our Summer Research Program that looked at the DNA damage response in an HSF1 mutant worm. This worm lacks a fully functional copy of HSF1. So as you can see on the left, the spermatocytes are showing very similar levels of DNA damage as before with this non-functional copy of HSF1. But on the right, now we suddenly see very high levels of DNA damage in the oocytes, comparable to what we saw in the developing sperm. So this was suggesting to us that not only does HSF1 provide a protective role against DNA damage, but it does so in a sexually dimorphic way, only protecting developing eggs against heat-induced DNA damage. So the next question that we were interested in exploring was, how exactly is this pathway led by HSF1 different between the adult male and the adult hermaphrodite? Or in other words, how is the gene expression changes that HSF1 is inducing following a heat stress different? And how might that lead to the sexual dimorphism and DNA damage response that we're observing? To address this question, we use a method called RNA-seq. The way this method works is normally in the cell, proteins are produced by the processes of transcription and translation, which produce this intermediate stage mRNA transcripts. And what we do is we collect these transcripts and we sequence them to identify which DNA genes they came from. And then we also quantify them in order to measure the extent to which each gene is being expressed or transcribed in the cell at a given point in time. So what this tells us is how much, this provides a measure of gene expression across a genome for a particular sample. For our experiment, we started out with plated worms. Males and hermaphrodites went through this process separately. We either exposed them to a 34 degrees Celsius heat shock for 2 hours or no heat shock as a control. We then took these experimental groups and we put them through the RNA-seq process in order to measure their gene expression levels. Following this, we took the gene expression levels seen from the heat shock groups and compared them to the non-heat shock groups. And this was so that we could identify which genes were being changed or expressed differently, significantly differently after a heat stress compared to a non-heat stress condition. So our results show that the male produces a strikingly larger number of gene expression changes following a heat stress compared to the hermaphrodite. So already this is pointing at an interesting sexual dimorphism in the heat shock response. But we're particularly interested in genes that are known to be targets of the heat shock response pathway as led by HSF1. So once we break down this gene list into just those known HSF1 target genes, this is what we see. It's very similar, as you can tell, proportions to the previous Venn diagram, with the males changing expression of about 5 times as many genes as the hermaphrodite within this pathway. And if we break it down into genes that are upregulated in their expression or downregulated, we see that the male is definitely upregulating a majority of these genes, whereas in hermaphrodite, it's more 50-50. So to further characterize the heat shock response in males and hermaphrodites, we decided to focus in on the expression of heat shock proteins. These are classic canonical targets of the heat shock response pathway, and they are involved in directly ameliorating the physiological effects of heat stress. So these are frequently used as markers of the heat shock response pathway or indicators. And when we look at the expression of these heat shock proteins in males and hermaphrodites, what we see is that in the males, they're upregulated by about 4-fold, whereas in the hermaphrodites, we see little to no change in their expression. So this is indicating to us that there is a sexual dimorphism in the response to heat stress by this HSF1-led pathway in males and hermaphrodites, and also that 2 hours following a heat shock, this pathway is more engaged in males than it is in hermaphrodites. Additionally, our study identified a few potentially interesting target genes that could be involved in DNA, that are involved in DNA repair and the maintenance of genome integrity, and could be involved in the DNA damage response that we see in males following a heat stress. These genes include XPG1, which is more famous in humans for its role in xeroderma pigmentosum. It's a DNA repair gene involved in converting different types of DNA damage to double-strand breaks, which is a particular type of DNA damage that's observed in the male-specific heat shock response. It also identified SPEC1, which is a gene involved in packaging DNA, and it's male-specific and also has roles in DNA repair. And also NERF1, which is a chromatin remodeling gene, which also has roles in DNA repair. So the big takeaways from this study are that following heat stress, males show a lot of change in HSF1 target gene expression compared to hermaphrodites, which show relatively little HSF1 target gene expression change. This is suggesting to us that there is sexual dimorphism in the HSF1 pathway, and also that, that it seems to be more engaged in males 2 hours following a heat shock than it is in hermaphrodites. Some future questions we're interested in pursuing include what does the HSR, the heat shock response, look like over time? We could do a time course analysis of HSF1 pathway activation in each sex after a heat stress, and we could use the expression of HSP70, one of these heat shock proteins I mentioned earlier, as a proxy for the whole response rather than having to do RNA-seq at each time point. And we're also interested in making use of C. elegans' great mutant catalog availability to look at how different mutants respond to heat stress in terms of their DNA damage response. And we're also interested in repeating this experiment using only germline samples, so that we're looking specifically at gene expression in developing sperm and egg cells rather than in whole worms. I would like to thank everyone in the Labuda Lab for their incredibly helpful support in focusing my ideas on this project, and also for substantially improving the presentation through their comments and advice. And I'd like to particularly thank my mentor, Nicole, who not only was able to spend significant amount of time and attention helping me through the sometimes challenging analysis of this data, but was able to do so on top of the additional challenge of remote mentoring. And I'd also like to thank the National Institutes of Health, the Knight Campus for Undergraduate Scholars, and the SPUR program for funding and support. I'm happy to answer any questions later, I guess.
Thank you, Phil. Yes, I will ask everyone to hold their questions until the very end. We have a full schedule. And so we will move to our next presenter, Deanna Plunkett, who will be presenting her project, Neonatal Inflammation Increases Adult Vulnerability to Subsequent Inflammatory Stimuli. Deanna, it's all yours.
Okay, perfect. I just want to say everyone is doing such an amazing job so far, and I'm very proud to be a part of this group. So I just want to start off with that. But as was just said, I'm Deanna Plunkett, and I'm working in the Huxtable Laboratory, and my presentation is Neonatal Inflammation Increases Adult Vulnerability to Subsequent Inflammation in Respiratory Control Regions. So neonatal inflammation is inflammation that occurs early in life, and it's actually extremely common, with up to 65% of preterm infants experiencing a bacterial infection while hospitalized. Unfortunately, this common early life insult has lasting consequences, like increased risk for psychiatric and neurodegenerative disorders. Additionally, neonatal inflammation impairs adult respiratory control, and our lab is trying to explore the mechanisms behind this impairment. One of the cell types that we think may be involved in this impairment are microglia. Microglia are the brain's resident immune cells, and in early life, or in neonates, microglia are in a pro-inflammatory state in order to aid with brain development. In adulthood, microglia are in a surveillance state, so that following adult inflammation, they increase in number in order to regulate inflammation. Following neonatal inflammation, however, microglia remain pro-inflammatory into adulthood, so that following subsequent adult inflammation, they have a greater increase in number. An initial stimulus leading to an augmented, often increased, or an altered, often increased response to a subsequent stimulus is priming. Microglia priming has been shown in other regions of the brain and has been associated with impairments in the outputs of those regions. For example, microglia priming in the hippocampus impairs learning and memory. Thus, microglia priming in respiratory control regions may impair respiratory control. The respiratory control regions of the brain are the medulla and ventral cervical spinal cord. The medulla is the primary region responsible for controlling the rate and depth of breathing. And it's the respiratory rhythm generator within the medulla that communicates to motor neurons in your ventral cervical spinal cord. The motor neurons innervate the diaphragm, and it's the contraction of diaphragm that allows air to enter the lungs, thus facilitating breathing. To explore if microglia are primed in respiratory control regions, we hypothesized that neonatal inflammation followed by adult subthreshold inflammation or a usually insignificant inflammatory stimuli will increase microglia in respiratory control regions. We chose to use an adult subthreshold inflammatory stimulus, as in healthy adults, microglia will not increase after this stimulus. Thus, if microglia increase after neonatal and adult inflammation, it demonstrates priming, but also suggests that adults who experience neonatal inflammation may be at a greater risk for, may be more susceptible to adult inflammation. To test this hypothesis, we dissected out those respiratory control regions, the medulla and spinal cord. We dissected out these regions so that we could immunofluorescently label our cells of interest, microglia. Microglia can be identified by their unique expression of the cell surface protein CD11b and CD45. Once our samples were immunofluorescently labeled, we could use flow cytometry to identify the percent of cells within each sample that were microglia. And we defined microglia as the cells that highly expressed CD11b and lowly expressed CD45. Thus, if microglia were to have increased, the percent of cells in this bottom right quadrant will increase relative to all cells within the homogeneous. We used this method first to actually establish an adult subthreshold inflammatory stimulus in respiratory control regions. To do this, we tested 10 doses of lipopolysaccharide, or LPS, ranging from 0.5 to 1,000 micrograms per kilogram. These low doses of LPS led to minimal or no increase in microglia, whereas larger doses led to maximal increases. And this dose response allowed us to determine the point where there's a 50% increase in microglia, allowing us to identify 24 micrograms per kilogram of LPS as a subthreshold inflammatory stimulus. That's 24 micrograms per kilogram of LPS in healthy adults will not increase microglia. After establishing the subthreshold inflammatory stimulus, we could begin our main experiments. To this, we had 4 treatment groups. Animals treated with neonatal saline are our solid bars, and those that received neonatal inflammation or neonatal LPS are striped. The animals were then aged and received adult saline as adult controls, or adult subthreshold inflammation, or the adult subthreshold dose of LPS. Neonatal inflammation followed by adult saline increased male medullary microglia. Further, neonatal inflammation followed by adult subthreshold inflammation further increased male medullary microglia. In other words, after neonatal inflammation, microglia increased in response to what would usually be an insignificant inflammatory stimulus, demonstrating priming and suggesting increased vulnerability to adult inflammation after neonatal inflammation. In the male spinal cord, despite no increase in microglia after neonatal LPS, neonatal and adult LPS combined increased microglia, demonstrating microglia priming in the male spinal cord. As well as these differences in microglia within region, we saw differences between region, as neonatal LPS increased medullary microglia more than it did spinal microglia. And this highlights that while priming occurred in both male respiratory control regions, there seems to be differences in how that occurred. In females, we saw slightly different results. As in contrast to what we saw in the male medulla, neonatal inflammation did not increase microglia, but microglia priming was demonstrated in the female medulla as neonatal and adult inflammation increased microglia. In the spinal cord, there were no differences between groups. Thus, microglia priming did not occur in the female spinal cord, in contrast to what we saw in the males and in contrast to what we saw in the female medulla. Because priming only occurred in the medulla, neonatal and adult LPS increased microglia compared to all spinal groups. Thus, neonatal inflammation primes female medullary microglia. So neonatal inflammation followed by adult subthreshold inflammation increased male medullary and spinal microglia, but only increased female medullary microglia. Thus, neonatal inflammation region and sex dependently primes microglia in respiratory control regions. Neonatal inflammation is a very common early life insult, and we know that it has lasting consequences. And our data suggests that one of them may be an increased risk for impairments in breathing control due to microglia priming in respiratory control regions. So adults who've experienced neonatal inflammation may be a vulnerable population who are potentially at a greater risk for breathing control impairments during adult illness. And I'd of course like to say some thanks of the Huxtable Laboratory for their continued support, specifically my PI, Dr. Huxtable, and my mentor, Sarah Baylor, for both helping me out considerably, my family for always supporting me, and there have been so many other people outside of the Huxtable Laboratory, my family, who have also assisted in this work. I'd also like to thank the funding that we received from the NIH in the Huxtable Laboratory to conduct this research. And I'd like to thank the Center for Undergraduate Research and Engagement for awarding me their Summer Undergraduate Research Fellowship, which allowed me to focus on research this past summer. So thank you. And I'll save questions for the end.
Great. Thank you for that, Deanna. And we'll move to our next presenter, who is Sunny Zhang. And Sunny will be presenting on magnetoelastic sensors for real-time tracking of MSC growth. Sunny, it's all yours.
Thank you. Hi, everyone. My name is Sunny, and in association with the Ong Lab at the Phil and Penny Knight campus, my research focuses on the development of magnetoelastic sensors for the real-time tracking of mesenchymal stromal cell growth. Cool. So most of us have heard about stem cell therapy. It is simply the promotion of the repair response of diseased, dysfunctional, or injured tissues using stem cells. Stem cell therapy holds immense potential in regenerative medicine, offering the opportunity to treat a wide range of diseases and injuries that persist in our society today. The use of mesenchymal stromal cells, or MSCs, in stem cell therapy has shown the capability in differentiating into mesenchymal tissues, such as muscle, fat, and bone cells, and have shown to have immunosuppressive activity. Stem cells themselves are like building block cells that have the capability to self-renew and differentiate into other cells with specialized functions. They are typically a part of our body's healing and developmental process, which makes them so intriguing in regenerative medicine. The past decade especially has seen immense research and funding into the industry. However, despite that, there's still so much more to be learned about stem cells. Current procedures and production of stem cells rely on identifying the key variables of cell count and viability and consistently producing optimal products. But despite being the gold standard in cell tracking, current methods can have some limitations in their accuracy and viability. MSCs are typically produced in bioreactors with suspension of microcarriers to aid in growth. This culture setting, although productive, does see limitations in certain tracking techniques for cell count. For instance, certain microscopy-based techniques aren't available for current methods of cell production because of the three-dimensional structure and optical properties of the microcarriers that these cells are grown on. Other methods, such as chemical-based assays, are able to be used to track cells, but often result in the destruction or disruption of these sample cells, and thus aren't a feasible method for tracking cell numbers. Traditionally, MSCs are counted using a multi-step process that involves trypsin digestion and trypan blue staining. This method has been shown to be viable in tracking cell parameters. However, it doesn't do so in real time, and it's time consuming with chances of procedural errors. So these limitations in all could lead to either a waste of MSC cultures or the production of unusable products due to procedural errors. And when stem cell research and production rely so heavily on consistent data and feedback, it is imperative to find methods in limiting the sources of errors and increasing accuracy. Magnetoelastic sensors, or ME sensors, are an alternative to current methods of cell tracking that can track key cell parameters in real time without disrupting or destructing cell samples. ME materials are magnetostrictive and thus convert magnetic energy to mechanical energy or vice versa, and this causes them to experience a physical deformation and mechanical resonance when exposed to a magnetic field. This property makes them an optimal medium for measuring a range of biological and chemical parameters. When a magnetic field is exerted onto them, the longitudinal vibrations produced by these sensors convert to elastic waves that generate a magnetic flux, which can be detected remotely, producing a resonance frequency. Any mass changes or loading on these sensors changes the resonance frequency that is produced. Even environmental changes of the cell cultures can affect sensor vibrations and be detected. The resonance itself depends on the system that the sensor is placed in, and so any changes is due to changes in the system. And this is pretty similar to the light sensors that we have today. In a room or a cell culture, for instance, any changes in the room result in the sensor to signify that there's something different about the room. This ability to respond to changes allow the material to not only detect and measure physical parameters like pressure, temperature, and liquid density, but also capable of chemical sensing. Current research on ME sensors and tracking biological and chemical parameters have only been done so on bacterium and virus populations, showing both plausibility and reliability in ME sensor tracking. However, very few research has really been done on living cells. It wasn't only until last year in which research conducted by the Ong Lab studied the use of ME sensors in tracking live mammalian cell culture growth of L929 fibroblasts. And through that study, it was showed the proof of concept that these ME sensors do have the potential in tracking live mammalian cell cultures in real time. However, it still remains unknown whether or not these sensors can actually track stem cells. And so the importance in developing a convenient and inexpensive tracking system using these sensors is to offer an alternative approach to tracking cell numbers rapidly with minimal evasion, offering both inline and in-process monitoring that reduces the probability of process errors. It's imperative within our studies to show the proof of concept for a system such as this so that we can further improve the developments in regenerative medicine and stem cell research. And so the goal of my research is to develop a detection system using ME sensors that can remotely detect mass changes in MSC cultures in real time with both accuracy and efficiency, as well as showing proof of concept for this. And through this, we also hope to extend this technology into creation of a feedback control system that can not only detect mass changes on the sensors, but also environmental changes within cell cultures. Currently, our procedures in tracking MSCs are by loading them onto a magnetoelastic sensor, which are then put into our detection system. The ME sensors themselves are fabricated from commercially available ME material, and they are further prepared by annealing and parylene coating them to make them more uniform in their surface area and biocompatible. They are also plasma etched to promote cellular adhesion because MSCs themselves are anchorage dependent. And thus, the sensor system is composed of a 2-coil system, a DC bias, and a sensing and detecting coil. The coil themselves were 3D printed by our own design and machine wound. This figure itself gives a great illustration of our intended mechanics and settings of the sensors. We plan on having the cells adhered onto cell microcarriers on the sensor themselves. And then we also plan on generating a magnetic field through the DC excitation to produce a resonance frequency that we can detect through our AC excitation and detection coil. Our data collection process is an extension of the procedures conducted last year with the L929 fibroblasts in our lab. We intend on extracting the frequency that the sensor generates and using that to calculate the impedance phase, showing the change in mass on the sensors. Our procedures in doing so are, again, very similar to those that were used with the L929 fibroblast. We intend on seeding the sensors at varying cell densities to produce different cell numbers and to test the effectiveness of the sensor detection system. And we will also be using sensors that aren't loaded with any cell cultures as a control group. On the screen, you can kind of see the results of our testing done last year with the L929 fibroblasts. Resonance frequencies were taken every hour for each cell density. And through that, we calculated the change in phase that would signify the changes in mass on the sensors themselves. And so you can see how the proliferation of the cells were tracked at every specific hour measurement. This data processing would be what we were also aiming for with the MSCs and what we would expect to see through this method and showing proof of concept. We're also planning on doing live dead assays on the cell samples using fluorescent dye and confocal imaging to evaluate the accuracy and create a standardization for the resonance and the sample procedures. So currently within our research, we've constructed our detection system with a DC-AC coil system as well as a detection coil system. Recently, we were supplied with primary MSC cells by CMAT and have undergone culturing of them. We've been able to get some preliminary live dead assays as shown here on the screen with the MSC cells. All of them are alive on this figure that we are presenting right now. We plan on having more developments and collection within the coming weeks, planning on loading MSC samples and varying densities on sensors to be monitored and using that data to find differences in resonance frequencies to obtain any changes in phase or changes in mass. As well, we're also planning on doing more live dead assays using confocal imaging for cell samples to verify that the cells we are observing on the sensors are both alive and are in real time. Within the coming weeks, we aim to have more supportive data that can validate our system of cell tracking and proof of concept that the sensors can track cell parameters and in real time and remotely. And finally, I just want to say thank you to all the faculty members at the Ong Lab, for Professor Ong, Salil Karapot, and William Skinner, all of them for helping me and mentoring me through this project. And thank you all for listening.
Okay, thank you, Sunny. And so now at this point, we will open it up to questions for everyone, for all of our 5 panelists who have been admirably punctual in their timing. So that leaves us ample time for questions. There is, as I've said before, an opportunity to type your questions in the Q&A box, or you may ask them verbally. Questions? I will give people a moment to formulate, come up with questions. So,
I guess this isn't research specific, but I am curious what everybody is doing in terms of like if everyone's pursuing graduate school to do research similar to what they're working on or switching fields or, yeah.
Thank you. Yes, Nora, I had the same question, so I'm glad you asked that. Who would like to begin? We can simply go in order. Carly, would you like to begin?
Sure. I think there are so many options that I haven't quite narrowed it down, but I definitely want to go to grad school, get my PhD, and then after that, who knows? I might stay in academia, I might go into industry. I think I have time to explore that, so I'm just excited to continue.
Great, thank you. Nora, would you like to continue?
Yeah, I'm actually going to do a master's in bioinformatics it'll take 18 months. And then after that, I'd like to do my PhD in bioengineering or molecular engineering.
Great, thank you. Phil?
I'm not sure which grad schools in particular, but I'm also interested in pursuing a PhD in molecular biology, molecular genetics, and maybe a crossover somehow with entomology.
Okay, great. Deanna?
Yeah, I feel a little like the odd one out here. I'm interested in going to medical school. So yeah.
Great, and Sunny?
Yeah, I am also interested in going to medical school.
Yeah, so not the odd one out after all. Great, other questions from the panelists or from those in the audience?
I had a question for Phil. I was wondering with the way that the heat shock, I was wondering like what that represents in like a human, like what kind of insult you were like replicating with that and like how common that is.
Right, that's a good question. So normally in mammals in general, for spermatogenesis to function properly, it has to function at about 2 degrees Celsius lower than core body temperature. And that's tying into the same thing. Things like, you know, hot tubs or a laptop on the lap, or that sort of heat is relevant. Studies have been done on people who work in like steel manufacturing industries and other industries where they're exposed to frequent heat that show a loss of infertility, similar to what we're seeing here.
Thank you. Other questions?
So Carly, over the summer, are you going to actually be like expressing some of the Affibodies you designed in lab?
Yes, I will be. And by that point, they won't actually be Affibodies anymore because Affibodies are those really particular like 3 helix structures. And so some of my structures will have helices, but they'll also have beta sheets. And then I'll be working with Jonathan to do flow cytometry on those.
Awesome, that's so exciting.
I'm excited. Maybe we'll see each other on the night campus.
Okay, this is the last opportunity for anyone to ask any questions. There is a question coming in over the chat, which I will read. It is a question to Deanna. I'm curious, and it comes from Alina or Elena Seligan. And I'm curious if you have a hypothesis on why there are differences in microglia priming between the sexes.
Yeah, really great question. And hi, Alina. Nice to see you. And so there may be a couple reasons for why there are possible sex differences. And I really think it's interesting to continue to explore, and it's something our lab is continuing to explore. So for one, during the timing of the neonatal insult, males have more microglia than females, and they seem to be sexually dimorphic in neonates in that aspect. We also know that in adults, healthy microglia are sexually dimorphic in their like metabolic activity and their inflammatory responses. So it's likely that there are underlying differences in already healthy microglia, which may contribute to the differences that we see after neonatal or adult inflammatory stimuli. But yeah, super interesting and likely has different consequences, or may have different consequences. So it's really important to explore. And I do want to point out, unfortunately, there's not as much research into females in the microglia field in general and within respiratory control, despite there being clinical differences in respiratory control disorders. So it's nice to see that as I've been doing research for this, more and more people are focusing on looking at both sexes.
Okay, we'll give one more opportunity for questions, either in the chat or live. Okay, if not, I want to thank all of our panelists for such a fantastic job, interesting projects, the difficulties coming along with doing research projects and presenting through Zoom and through our socially distanced time. So congratulations to all of you. That's fantastic. It's also, it sounds like you have interesting projects going on after this. So thank you all very much. Congratulations on this. And thank you for your presentations.
Thank you for moderating, Cory.
My pleasure.